Hi 1409 students. Today we're going to talk a little bit about the kingdom Animalia within the domain Eukarya, and there is a lot of diversity in this kingdom. I've just put a couple of different pictures here on the intro page to have you see the difference between vertebrate and invertebrate organisms and hair and feathers and all those kinds of things. So that's what we're going to take a look at in this PowerPoint. Okay, now I've loaded I think this is a really good intro to animals. It's one of the TED Talks from David Gallo. I'm not going to play it right now, but that might be something that you want to watch before we start looking at all of the different characteristics of animals. So I've listed a few here for you to fill in, and we've talked about a lot of these things already, but so that you have your list. Everything in the animal kingdom is going to be heterotrophic, and heterotrophic by ingestion, meaning that food is taken in, digested inside the organism, and then the nutrients are used for energy and so forth. Every organism in this kingdom is multicellular, so there are no single-celled organisms, they're all multicellular. Most of them have tissues. So we talked a little bit about biological organization in 1408, and we're going to talk about it again in 1409 when we look at all of your body systems. But we're all made of cells, and if you put a bunch of cells together and they all work together to do a particular job or function, we call that a tissue. And so most animals will have tissues and organs. There are a few that are lower that don't have true tissues. Okay, typically move, so that doesn't necessarily mean every part of the life cycle they can move, but at least in one part of their life cycle, animals will be able to move. For reproduction, all animals can reproduce sexually, so that means there's input of genetic material from two parents. They all go through a development process, and during this development process, most of them will produce a blastula. So we're going to talk a lot more about blastulas when we get to reproduction and development, but I'm just going to, let me see if I can draw just a little bit of a picture on the screen here. But remember, during development, we have an egg and a sperm that get together, fertilization, and that makes a single cell, which is a zygote. Okay, and then this zygote goes through many divisions in the process of development. But at the very beginning, you have a solid ball of cells, and then that solid ball of cells starts to push the cells out, and the blastula stage is where you have your ball of cells, but now all the cells are pretty tiny and they're all pushed out to the edge. So I'm not going to be super specific on this test for what a blastula is, but I want you to understand it's a step in development. Okay, and so when organisms go through development, we can either talk about direct or indirect development. And so I'm seeing that my picture is kind of covering up a little bit of the bottom of this diagram. But if we look at letter A here, this is what would be considered direct development. So you have a juvenile that as it grows and develops becomes an adult, and they look about the same. They have all of the same parts. Growth and development just causes the organism to be bigger. In indirect development, you have an intermediate stage, which is the larval form, which does not look anything like the adult form. So you're going to have to look, I don't know how to move my picture over, but you're going to have to look at the PowerPoint to see the adult. But in metamorphosis, if you think of like butterfly development, we have a caterpillar, which is the larval form that does not look anything like the butterfly. That would be considered indirect development. Okay? All animals do not have cell walls. The outer boundary of their cells is a cell membrane, and all animals are aerobic, meaning that they require oxygen. Okay, so those are basic general characteristics of animals. And so now we're going to look at some of the characteristics that we would use to help us classify and decide where these organisms are going to go in our classification scheme. So the first thing you want to look at is how the body is put together and what kind of symmetry it shows. Okay, so if you have an organism that is symmetrical, then that means that you're going to at some point be able to cut the organism in half and get to the both sides are the symmetrical or the same. Okay, so the lowest level would be an organism that does not have symmetry. It's asymmetrical. And so, for example, sponges we're going to look at as one of our first organisms. Sponges are asymmetrical. There is no way that you can cut an organism in half and get two sides that are the same. That is indicative of a very low-level organism. Okay. The second type, radial symmetry, means that the organism is symmetrical about a point, which means that we could cut it in half this way or this way or this way, and we would get two halves that are the same. That's also a somewhat lower level organism. So this is showing a hydra, which is in the same category as jellyfishes. Okay, and then the top level is bilateral symmetry. Okay, and in bilateral symmetry, you are symmetrical about a line. And so there's only one line that you can look at, that you can run down the center of that organism, cut it in half, and you're going to have two symmetrical halves. So if we look at this crayfish, we cannot cut it in half this way because the front and the back are different, but we can cut it down the middle. Okay, so we look first for symmetry in organisms when we're classifying. And then we look for cephalization. So cephalo, this word root here, means head. And so we are looking to see, does an organism have a head end and a tail end? So any accumulation of nervous tissue is considered cephalization. This one here is a flatworm. It's a pretty low-level organism. But it has a bunch of nervous tissue on one end, which would indicate that it has a head end. If we look at a butterfly, also has a head end. It's a little bit more complicated. It has mouth parts and eyes and antennae, and so that's a little bit higher level organism with more cephalization. Body plans, digestive system. There's a lot of different terms here that we could use. I'm going to use these two terms to describe the digestive system. So the lower level one is considered a gastrovascular cavity, and this is a digestive system that has only one opening, meaning that food comes in that opening, things get digested, and waste comes back out the same opening. You can probably understand that that's going to be a lower level organism. So this here is a flatworm, and this is the opening here. And so food is taken in there, goes through the digestive system and gets digested. Waste comes out the same opening. Okay? An organism that has a complete digestive tract is going to have two openings. Okay? So in this earthworm, there's a mouth. Food comes in through the mouth, it goes through the digestive system, things get digested, it uses the energy, and we can't see the tail end of this earthworm, but the other end is the anus, which is the end where all of the waste would be removed from, and so that's a higher level digestive system. So I mentioned the definition for tissues at the beginning. And so we look for true tissues in organisms, and then we look for germ layers. And this is something that we haven't talked about yet. Germ referring not to germs like bacteria, but germ referring to germinal, which means beginning. Okay? And so when an organism is going through early development, there are layers in that early ball of cells, and some of those layers are going to go to become nervous system, and some of those tissue layers are going to go to become skin, and so forth. So usually we see either 2 germ layers indicating a lower level organism with less organ systems, or we see 3 germ layers. And so we'll be more specific about that when we get to reproduction, but that's something that we look for in organisms to determine how to classify them. We also look for segmentation. Segmentation means that, well, you see these different body parts, and if you see segmentation, that indicates that the organism is a little bit higher organism because it's got different body parts that do different things. So if we look at the lobster here, obviously it's segmented, and you can see that we have different segments at the front that have different functions, and we have some more segments in the middle, and we have different segments at the end. That's specialization. Different parts of the body do different things. This caterpillar is also segmented, but a little bit lower organism, because you notice that all of those segments are the same. So there's not as much specialization in a caterpillar. Again, this is one of these things, I keep saying we're going to talk about this in reproduction and development, but we have to at least mention it here, and then we come back and talk a little bit more about it in a little bit more detail. But during embryonic development, and maybe I'll just draw a little bit of a picture here. So we said that sperm and egg get together and you make a zygote, okay? And then this zygote goes through a lot of divisions, and I'm skipping some steps here, but we get a blastula, which is made up of a lot of little tiny cells. So that zygote has divided and divided and divided. You have lots of cells, and this is what we call the blastula. So this is not looking like a real organism yet. Okay, and then we go through even more divisions and more growth, and we start to get this blastula. It's kind of punched in here, so kind of punches in right there. It's called a gastrula, and this will be the first opening for a digestive system. Okay, so the difference between a protostome and a deuterostome in terms of development is if this first opening becomes the mouth, then that's going to be what we consider an organism that is a protostome. If that first opening becomes the anus and the second opening becomes the mouth, that is what we call a deuterostome. Okay, so again, you don't have to know a whole lot about this right now, but understand that development is going to help determine what kind of organism we have, and they don't all develop exactly in the same way. Okay, so I drew a little picture, but here you can see, and again, I don't know how to move my picture over, but if this bottom picture is the protostome, and so the first opening down here became the mouth, and in the deuterostome, the first opening became the anus, and the second opening became the mouth. And so you're not going to have to diagram this or anything like that, but there's a difference in the way these organisms develop. Okay, and then we are going to look at the difference between organisms that are invertebrates and organisms that are vertebrates. Okay, so most animals are invertebrates. You don't have to memorize numbers here, but 95% is a big percentage. Okay. Of all of the animal species that have been described, which is in the millions, only 57,000 are vertebrates and the rest are invertebrates. So that's a lot of organisms that are in this category. And maybe some of these are ones that you're less familiar with because they're not as big as vertebrates. So we are going to spend the first half of the animal chapter looking at the invertebrates and the second half looking at the vertebrates. Okay, so I'm not worried about you being able to label a chart like this, but I like it for representation. Okay, so down here at the bottom, we said that if you're going to be in the animal kingdom, you're going to be multicellular. Okay, then we said we're going to look to see if you have tissues or not. And so you notice way over here on the left, sponges don't have tissues. These are the oldest and the least developed, or the least, what's the word I'm thinking of? Complicated organisms. They're very simple organisms. Okay, then we look at the symmetry. Okay, so we talked about asymmetry and radial and bilateral symmetry. So everybody on this side has bilateral symmetry, but the cnidarians, which we're going to talk about, jellyfish are in this category, have radial symmetry. So they're also on the lower end of the scale. Okay, then we look at the organisms on whether they're a protostome or a deuterostome, which we just talked about those definitions. So everybody on this side is a protostome. And over here on the right, these guys are deuterostomes. I am not worried about these. I'm not going to use those terms. Okay? And so then when we look at all of these organisms, we're going to look at, well, what's their circulatory system like? What's their digestive system like? We're going to look at those kinds of things. We're going to look at segmentation, and that's going to help us determine what group to put these organisms in. So I like this diagram. It comes from another book, but I like it. Okay, this one is in the McGraw-Hill book, and it's basically showing you the same thing. It's just tipped sideways, but you have the organisms going from the simplest organisms to the most complex organisms, and then you're looking at these different characterizations that we're trying to determine. Okay, and again, I'm not going to worry about these two right here. Also in your book, you have a table that lists all the phyla of animals in the animal kingdom. It lists examples. It gives numbers of species. For sure, I'm not interested in you memorize anything about numbers, okay? But it's kind of a nice table that lists each phylum, and we're going to talk about these phylums, and we're going to look at some pictures and some videos. So it's kind of a nice review once we finish looking at the different groups of organisms in the animal kingdom. So we're going to start at the bottom. Remember, we said the sponges are the least complex or the most simple organisms. The phylum name is Porifera, meaning pore-bearing, or it has pores, which pores are holes, right? Excuse me. And these organisms are asymmetrical. So I'm showing you a few different pictures here, but none of them are perfectly round or can be cut in half down a line, and so they're asymmetrical. Very big difference in size and shape. There are some that are almost microscopic and you need a microscope to look at, but there are some that are 5 or 6 feet tall, okay? During their adult life cycle, they are non-modal or they are sessile, meaning they don't move. You do see movement in the gametes, but that's the only movement we see in sponges. I've lost my cursor here. I guess that's okay. So a couple of characteristics for the poriferans, the sponges. They're filter feeders, meaning that they're filtering water, and so they do have these pores within the body wall that water will go through. They filter out food from the water, and then the filtered water comes out the top here, okay? Every animal has to be multicellular, so sponges are multicellular, but they don't have tissues, meaning that every one of their cells is doing its own thing. They're not working together as a tissue. They don't have a gut. They don't have a digestive system. So basically, when water is filtered and food is pulled out of the water, it's digested within individual cells, but there is no digestive system. So it's very simple. And they can reproduce both sexually and asexually. So asexually, a part can be broken off and it will grow into a whole new organism, but they can also produce sperm and eggs. So just a couple of different pictures of sponges. You can see different sizes, different shapes, different colors. I added a video here on sponge reproduction so you can see their production of sperm and egg and how that happens in the water. And then just for perspective here, a couple of questions to think about. So minerals in sea sponges. And we're going to talk about some of these minerals as being important in digestion when we get to that in human digestion. But in terms of minerals that you find in the sponge, if you haven't read through this already, think about the minerals and maybe think about what you might find in the ocean. And then second question, if you were to break a sponge in half, what would happen? So if you took a sponge and pulled it apart into two pieces, would it die? Would it be able to reproduce? Would it form back into another sponge? Would it become two sponges? That's something to think about. So I'm going to go ahead and click on the answer. So iodine is present in a lot of marine organisms in the water, and iodine is one of those really important minerals for humans for metabolism. So that's an important one. Most people don't eat sponges. That's not usually a food item, okay? And then if you were to break a sponge either in half, or you could really even take a sponge and squish it through like a colander or something where it breaks it apart a little bit, and then all the pieces you collect in a bowl. Basically, those will all come back together and you'll reform that sponge. So that means this is a very simple organism. If you can pull it all apart and it's just going to coalesce back together and become one organism again, there's nothing like that in higher organisms. We could never do that. Okay?